Hi everyone, today we'll be covering the Pearson Edexcel IGCSE Human Biology paper. So this is from 2023 January and this is paper 2. So for this paper, you're given an hour and 45 minutes to complete and the total mark for this paper is 90. So this paper tests on the entire syllabus. Okay, let's start off with question 1. The image shows part of the skeleton. Label the clavicle, scapula, and vertebral column on the image. The clavicle is what we refer to as our collarbone, so this bone here would be the clavicle. The scapula is our shoulder bone, so this is the scapula. And lastly, the vertebral column, this is this one here. Vertebral column. Okay, part 2. The vertebral column is part of the axial skeleton. Describe the functions of the axial skeleton. Okay, so before we start, for part of your syllabus, you need to know two types of skeletons. Let me write this down. So we have the axial skeleton, and we have the appendicular skeleton. So your axial skeleton, that consists of your skull, your vertebral column, your rib cage. And that's mostly important for protecting the body, or protecting internal organs specifically. And your appendicular skeleton, which consists of your scapula, your clavicle, so these two here, as well as the pelvis and the limbs. The appendicular skeleton is responsible for movement. So going back to this question, describe the functions of the axial skeleton. Well, number one, it's there to protect your internal organs and also to support the body as well. So the function is to support the body and to protect internal organs. Okay, and sorry, I forgot to mention, I've included this little image here which shows the axial skeleton. Let me just make this one larger. So for your syllabus, you'll need to know the skull, the vertebral column, and the rib cage. All right, let's move on with the next question. Part B, osteoporosis can affect the vertebral column. Describe the symptoms of osteoporosis. Included a diagram here which shows the difference between a normal bone and a bone with osteoporosis. So on the right, we have osteoporosis and we can see that it's a lot more porous, right? And so there's an overall decrease in the bone mass. And if it's a lot more porous, then it's more likely to break or more likely to fracture. And that could affect the person's posture and also cause an immense amount of pain as well. So let's just write that down. So we have to talk about what osteoporosis is. So osteoporosis leads to the bone becoming more porous. This leads to, or this can cause perhaps, this can cause the bone to fracture easily. The person would experience pain and may have a poor posture. So people with osteoporosis tend to be kind of hunched over. Okay, let's go over the marks. So becoming more porous, that's one mark. Bone to easily fracture, that's a second mark. And poor posture is a third mark. And pain is another mark that you can say. All right, and let's move on to question 2. The diagram shows a section through the heart. Part 1 says name chamber Y. So let's name both of these structures first. So this is the right side of the heart, and this is the left. So structure X here, this is a valve. So the valve prevents backflow of the blood. And then chamber Y here is the left ventricle. So actually, let's write that down here. So name chamber Y, that is the left ventricle. Now, explain the function of structure X. So we mentioned earlier that structure X is the valve. So what would happen is as blood gets pumped from the atrium to the ventricle, when the ventricles contracts, it pushes blood upwards, right? And when that happens, the valve prevents blood from going back in to the atrium. Okay, so let's write that down. So structure X is a valve, and it prevents the backflow of blood. So one mark for naming what it is, and another mark for prevents backflow of blood. So that's 2 marks. All right, let's look at part 3. The table lists some properties of blood. Add ticks to those boxes. Let's show the properties of the blood in the aorta. Okay, let's go back up here. So this is the aorta. Now the aorta is carrying oxygenated blood. So here we have this is the pulmonary artery. So this pulmonary artery would pump blood to the lungs, and then it would go down to the pulmonary vein, and then it would go to the aorta. So your aorta would have oxygenated blood. So let's see if we can tick that box. Okay. So properties of blood: oxygenated, that is true because it just came from the lungs. And then so it's not deoxygenated. It does have high pressure. So the reason why it has high blood pressure is because it's coming out of the heart, and the ventricles here are pushing the blood out of the heart. So the blood would be traveling at higher pressures. And yeah, that's part 3 done. So part 4, it says state where blood in the left side of the heart has come from. Okay, so let's go back up to this picture of the heart. So this here is the left side of the heart, right? And we just mentioned that earlier that the heart would pump blood to the lungs, and then blood will go back to the heart via the pulmonary vein. So it just came back from the lungs. So let's say the lungs. All right. Part B, in 1 year in the United Kingdom, there were 173 heart transplants. Heart transplant operations require hearts to be donated. Some hearts cannot be used because the donor is too old. So give 2 other reasons why a heart might not be suitable for a transplant. There's a few things that you can say about this. So whenever it comes to any form of transplant, you want to make sure that the organ matches the recipient so that you would decrease the likelihood of that organ being rejected. So we can write that down. So the heart might not match the recipient, which could lead to rejection. And Another thing you can also say is that the coronary arteries of the heart may have fatty deposits, and that wouldn't be very suitable for you to transplant that heart into a patient. So you can say the coronary arteries, arteries may have fatty deposits. Some other points that you can include is when you remove the heart from the person, the heart might be out for too long and therefore you can't transplant it into the recipient. Another point that's also in the marking scheme is that perhaps it's the wrong size as well. Okay, let's look at part C1. Which blood component causes blood clotting? So that would be the platelet, so that's C. Okay, let's just go over the blood components together. So both your lymphocytes and your phagocytes, these are your white blood cells. So they're responsible for killing pathogens in our body. And then we have our red blood cells, which are responsible for transporting oxygen around our body. Okay, so part 2. The diagram summarizes part of the process that causes a blood clot. Anticoagulant drugs prevent a blood clot forming. These drugs stop the enzyme thrombin, so this is the enzyme here, from being produced. Describe how a lack of thrombin prevents a blood clot forming. So if we don't have thrombin, what would happen is that your fibrinogen cannot be converted to fibrin. And when that happens, then your fibrin can't be converted to a blood clot. So that's what we can say here. So yeah, we can write here, fibrinogen would not be converted to fibrin, and therefore the fibrin cannot form the blood clot. Okay, so those are the 2 marks. Let's move on to question 3. Student investigate diffusion using agar cubes and an acid. The agar cube contains an alkali and a pH indicator. The indicator is pink in alkali and colorless in acid. So if you guys take chemistry, you'd know that this is actually phenolphthalein. Acid diffuses into the cube, causing the cube to change from pink to colorless from the edge. Okay. So part A, the cube the student placed in the acid has a side length of 2 millimeters. So actually, let me just draw a little cube here. So each side is 2 millimeters. Calculate the surface area to volume ratio of the cube. So the surface area, so to find out the area of one side, we would do 2 times 2, right? And because there are 6 sides in the cube, we would times that by 6. So let me just use the calculator. That'll be 24. And the area would be 2 times 2 times 2. So 2 times 2 times 2, that'll be 8. Now, if we're talking about the surface area to volume ratio, that's 24 to 8. Well, let's divide both sides by 8 so we can simplify the ratio. Here we have 1, here we have 3. So that'll be 3 to 1. Okay. Part 2. The cube took 1 minute and 32 seconds to become colorless. Which of these show the time in seconds? So we know 1 minute is the same as 60 seconds. So we'd do 60 seconds plus 32, and that would be 92. All right, state what is meant by the term diffusion. So this is a common question that shows up. Sometimes they might ask you what diffusion is, or what osmosis is, or what active transport is. So you do need to remember the definitions for these words. So state what is meant by the term diffusion. That is when substances move from high concentration to low concentration. So for example, if I were to spray perfume in the air, the perfume would diffuse throughout the room. It won't just stay in one spot of the room. That would be a little funny. Okay, and then part 4. Explain the effect of increasing the surface area to volume ratio on the time taken for the cube to become colorless. If we increase the surface area to volume ratio, the rate of diffusion increases. And the reason why is if you have much greater surface area, then there is more area in which diffusion can actually occur. So let's write that down. As the surface area to volume ratio increases, so it's asking for the time taken, right? The time taken would decrease. So this is because there is a greater area in which diffusion can take place. Okay, let's move on to part B. The student wants to investigate the effect of temperature on the rate of diffusion into the agar cube. Describe a method for this investigation using the apparatus shown in the diagram. So here, because it's only 2 marks, you probably need to mention the independent variable and dependent variable. So independent variable is the thing that we change. In this case, we want to measure the effect of temperature. So that will be your independent variable. Your dependent variable is the rate of diffusion. So we can do this by, let's say, timing how long it takes for the cube to become colorless. So that would be how we would test for the rate of diffusion. Okay. Here's a range of different let me just double check this. Okay, so what's happening is that the acid is diffusing into the cube, so we want to change the temperature of the acid. So we could use a range of different temperatures of acid, so that's 1 mark, and then measure how long it takes for the agar cube to turn colorless. Okay. Part 2, give 2 variables the student would need to control in their investigation. Okay, because the independent variable is the temperature, so that means that some things that you would need to keep constant is the surface area to volume ratio of your agar cubes. So the surface area to volume ratio of the agar cubes, that's one thing. Or if you want to put it simply, you can say the size of the agar cube needs to be the same, or the volume of the agar cube needs to be the same. Either way, that would get you that mark. Okay, so another thing that needs to be controlled is the concentration of the acid. So concentration of the acid. Other things you might want to control is also the concentration of the indicator that is in the agar cube. Okay, let's move on to question 4. When blood flows into a network of capillaries, tissue fluid is produced. The tissue fluid supplies the surrounding cells with molecules. The diagram shows a network of capillaries and the surrounding cells. Give 2 differences between the composition of tissue fluid and the composition of blood entering the capillary network. So what's happening here is that this capillary here is carrying oxygenated blood, and then when it leaves, it carries more deoxygenated blood, and that's because what's happening is that the oxygen is diffusing out, and the carbon dioxide is actually diffusing in, because remember, I don't know if you can see that, but I wrote CO2, because remember your cells are undergoing respiration, so it requires oxygen and it's going to produce carbon dioxide. So one of the differences between the blood entering the capillary network and the tissue fluid is that the blood in the capillary network has more oxygen. So blood entering capillary or entering the capillary network will have a higher concentration of oxygen, so that's one mark, and then we can say it has a lower concentration of carbon dioxide. So entering the network will have a lower concentration of carbon dioxide.
Another thing you can also say is that there are no red blood cells or large proteins in your tissue fluid. And that's because your red blood cells and your large proteins are too large to fit between the pores of your capillaries.
Okay, let's move on to part 2, describe how tissue fluid is formed. So I've included this diagram here so you can see. So here we have the arteries on top and then the vein at the bottom. So your artery contains high blood pressure, and that actually causes the plasma to get forced out of the capillaries. And between the cells of your capillaries, you have a lot of pores that enable the plasma to actually come out. So things like water are able to leave the capillaries, and you can see that there. And even smaller molecules as well. But larger molecules and even like red blood cells, for example, they are not able to leave the capillaries unless there's some sort of bleeding, which we don't want. Okay, so we can try and attempt this question. So let's write this down. Tissue fluid is formed because the artery end of the capillaries have higher blood pressure. This causes the plasma to leave through the pores in the capillaries. So you'd also need to mention that water is able to leave, and so can small molecules. So small molecules, including water, are forced out of the capillaries. Okay, so let's go through the marks. We have higher blood pressure in the artery, that's 1 mark. We have capillaries have pores, that's the 2nd mark. We have small molecules and water. So small molecules including, I forgot to write water. So including water are forced out of the capillaries. That is the 4th mark. Okay, let's move on to part 3. Okay, lymphedema occurs when tissue fluid builds up, causing swelling. This is more common in the legs and arms. One treatment for this is to increase physical exercise or physical activity, resulting in more muscle contraction. Explain why increased muscle contraction could reduce the swelling caused by the buildup of tissue fluid. Okay, so actually let's bring back that diagram over here. So we have all of this tissue fluid that's like around the capillaries. And what would happen to the excess tissue fluid, and you see this here? The excess tissue fluid gets drained into the lymphatic system. And what does that have to do with muscle contraction? Well, muscle contraction helps move all of this fluid into that lymphatic system. So we can write that down. Let's just make this one smaller. Okay. So muscle contraction would help drain the excess tissue fluid to the lymphatic system. All right, that's your 2 marks. Okay, and then part B. Lymphocytes are found in lymph nodes. Lymphocytes found in lymph nodes produce antibodies in response to specific antigens. Explain why the reaction between an antigen and an antibody is specific. If you guys recall, antibodies are produced by lymphocytes, and they would specifically target and bind to antigens that are found on the pathogen. So I've included 2 diagrams here, but we're going to look at this one first. So this is the structure of an antibody. So this orange part here is going to be the same for all antibodies. Now this red part here is what we call the variable region. So this part is the one that specifically binds to the antigen. And I've included another diagram over here on the right. Let me just make this one larger. So for example, it's a little bit blurry, but if you have these A antibodies, these A antibodies, they're only going to recognize the A antigen, for example. Let me just bring this back again. So these A antibodies are only going to recognize these A antigens that are found on the pathogen. Likewise, these B antibodies are only going to recognize and bind to the B antigens. And this type of binding is what we refer to as complementary. So this shape is complementary to this shape here. Likewise, this shape here is complementary to this shape here. Okay, so let's make these diagrams smaller so we can write down the answer. The antibody contains a variable region which allows it to bind specifically to a antigen. The shape of the antibody is complementary to the shape of the antigen. Okay, so the 3 marks are variable region, that's 1 mark. Another mark is will bind specifically to an antigen. And then the other mark would be complementary to the shape of the antigen. So that's a 3rd mark. Another mark you can also say is that each antibody would be different. So they would all have very different variable regions. Okay, we'll move on to question 5 in the next video.